So let us now look at what is StringDB. And StringDB is a great resource related to protein-protein interaction networks. So what you see is more than 2,000 organisms are represented. You have 9.6 million proteins and 13.8 million interactions. Okay, and it is a European-based thing, so it's been sponsored by EMBL. We know EMBL because Ensemble, the genome browser, is an EMBL product. The Swiss Institute of Bioinformatics and the CPR, Center for Protein Research. Okay, so you can log in, you can register, you can store information. Let's just do a search. And what protein name are we talking about? Let's call actin. Actin is a muscle protein, and you can auto detect, but just keep it simple. Let's just go with Homo sapiens and select Homo sapiens, and let's do a search. Okay, and so you have lots of these links, and for actin related to actin, for example, just take one. CALD1 is caldesmin-1 actin and myosin binding protein implicated in the regulation. So you have many, but just let's look at one. And what you see here is a string which talks about the protein-protein interactions, and you'll see various proteins associated with this. Now you can do this to make things very simple. So there's kind of a nice technical thing that's associated with this. You can click on this particular link, and it'll tell you what is this protein. It's the villen epithelial specific calcium regulated actin modifying protein. So what you have is the various resources, and remember, you see a pattern beginning to form, right? That every time you have a resource, it also becomes a kind of a knowledge management. It becomes a more comprehensive knowledge resource. So for this protein, the villen-1 protein, you see you have a link to that protein in Ensemble. Gene Cards, which is hosted at the Weizmann Institute of Science in Rehovot, Israel. The KEG, the Kyoto Encyclopedia of Genes and Genomes, and we looked at that when we looked at the David resource. This is the NextProt resource and the UniProt resource. It tells you, gives you information related to families of proteins, for example. It tells you, the different domains, different motifs associated with this protein. It tells you about the homology model of the protein. For example, the homology model of different domains of this protein, and you can see there are several different homology models that are created. Right, and so these are homology models of the different domains that are created. If a protein PDB structure is available, for example, you see this is 3FG7, and you can go to, for example, PDB and look for 3FG7. And you will get that. You can see the villen domain 6, right? That was the 6th structure. And once we look at, once we begin to look at PDB, we'll begin to see this a little more. Okay, so you can see this is 1, 2, 3, 4, 5, 6. This is the structure of the domain domain number 6 in this protein. And so you can get similar information for all of these. Now let's see what the color code is, right? So the VIL1 protein interacts with the TNP1, it interacts with the ACTA1, it interacts with the ACTG1, and so this is like a pivotal central protein. Additionally, you'll see that each of these proteins also interact with other proteins. Okay, and so StringDB is really important because you see, well, this is a pivotal protein. You can kind of drag this and say, now in this particular interaction, you'll realize that the COMD6 protein or the COMD domain containing 6 has no resource information anywhere else other than in the EMBL resources, and so you can actually run a smart sequence analysis, this resource is available too. So wherever these instances of information are available, you can take a look at this. Now, in terms of this domain, you'll realize that this is the only one which has a protein-protein interaction with the Will1 resource, but it has nothing related to, it does not interact in any way with any of the other proteins. Okay, so let's look at what the color code is. The query protein and the first shell, so this is the first shell, this is the pivotal central protein, and the search that we did was based on that particular protein. The white nodes are a second shell of interaction, and if you see, if it's filled, see that if it's filled, you'll see that some 3D structure is known or predicted. When you say known, it probably has a link to the protein data bank. If it's predicted, which means there's a homology modeling associated with it. Now let's look at these known interactions. Now the interactions are color coded, right? So for example, this interaction between the Will1 and the COMD6 or the TNP1 are this light green, and light green means it's a result of text mining. Okay, so you can realize that in order to put these bits of information together, so there is no experimental information or based on information from databases, but somebody in some text in an abstract perhaps indicated that there was an interaction between the Will1 and the COMD6 domains, and so this tells you if there was a black line between any of these, and we can see whether there is a black line, it'll tell you, it means that the proteins are co-expressed. So these proteins, anything that is associated with a black line is co-expressed, and the fact that it also has a yellow line tells you that it comes from text mining. Anything that's red, and there aren't any reds here, but there's these interactions, which is magenta color, dark pink, for example, comes from experimental results. So that's experimentally determined. If there's a blue line, which I don't see here, it means that that these interactions have been placed in a resource related to these interactions. And if there is a kind of a light purple line, gene co-occurrence dark blue, which we don't see here. And so you get a lot of information related to all the protein. It tells you a little more. It tells you, and these are the other proteins that are in this particular network. It comes from the NCB taxonomy ID 9606. There's nothing new about that. It's just that the taxonomy ID for Homo sapiens. And so, for example, you'll see, for example, these dots, which tell you similarly how these come about. And notice we identified a lot of these interactions in this particular matrix that arise, that connect to Will1 via text mining. And so you can see that these are text mining, and this tells you the score in terms of the probability or the efficacy of identifying that particular interaction. So, you know, just because it's in there doesn't mean it's perfect. There's always probabilities associated with this. So this is information related to StringDB. And that's about all I have to say. You can look for other information like analysis, clusters, look at various resources. Once again, just like everything else, you know, you're being introduced to this resource, but if you have, if you did experiments, have more intimate knowledge from experiments, from your work in this particular field with a particular protein, you will get a lot more out of it. But what you have to understand is such a resource is available for you if you want it.